Welcome back to the deep dive. Today we are diving into the first time ever permanent genetic engineering is being tested inside live humans.
Yeah, it is a huge milestone for medicine.
It really is. I mean, imagine a person who eats highly processed fast food every single day.
Right, someone who never exercises and has a totally stressful lifestyle.
Exactly. And yet, when doctors look inside their body, they find the pristine, perfectly clear arteries of a healthy teenager.
Which just sounds impossible.
It does. And for decades, the medical community just assumed these rare individuals were wildly, inexplicably lucky.
Just genetic lottery winners, basically.
Right. But today we are talking about how scientists finally hunted down the exact biological typo in their DNA that makes them practically bulletproof to heart disease.
And more importantly, we are exploring the reality that for the first time in human history, we are actively, permanently editing that same protective typo into the rest of the human population.
Which is just mind-blowing to think about.
It is a monumental shift. I mean, we are looking at a transition away from the era of chronic, lifelong disease management.
Where you are just tethered to a pharmacy for decades, constantly taking daily pills.
Exactly, just managing symptoms. We are moving toward an era of one-time permanent genetic cures.
So our mission for this deep dive is to examine the science making this possible, look at the breakthrough clinical trials from this year, and confront the massive ethical and economic shakeups this brings.
Because when you introduce a potential genetic cure for cardiovascular disease, which takes about 19 million lives globally every year, you aren't just making a new drug.
Right. You are fundamentally altering human longevity.
Precisely.
So before we can talk about how we are rewriting the human genetic code, we have to understand what exactly we are trying to rewrite.
We need to know why our bodies need fixing in the first place.
Right. And most of us have a vague understanding of this. You go to the doctor, they run a blood test, and they tell you your bad cholesterol is too high.
Which is a very common experience for a lot of people.
Yeah, but what is actually happening inside the pipes of the body that requires a genetic intervention?
Well, to understand the fix, we have to look closely at how the body processes fats in the blood.
Okay, break that down for me.
So cholesterol itself isn't actually evil. Your body needs it to build cells and make hormones.
So we need some of it to survive.
Exactly. But cholesterol is a fat, and your blood is mostly water.
And oil and water don't mix.
Right. So the body packages this fat into tiny protein vehicles so it can float through the bloodstream.
Like tiny boats carrying cargo.
That is a great way to picture it. The problem is when there are just way too many of these specific bad cholesterol boats floating around.
They start causing traffic jams.
Worse. Over time, they crash into the inner walls of your arteries, get stuck, and trigger a massive immune panic.
Which causes inflammation.
Right. And that builds up into plaque, which basically clogs the pipes. If that plaque breaks open, it causes a clot, completely cuts off oxygen, and boom.
A heart attack or a stroke.
Exactly.
So if having too much of this bad cholesterol in the blood is what clogs the pipes, how does a healthy body normally clear it out?
There is a built-in filtration system, and your liver is the main vacuum cleaner.
Okay, so the liver is the vacuum.
Yes. The surface of your liver cells are covered in thousands of microscopic biological handles.
Like little attachments on the vacuum hose.
Perfect analogy. As blood flows through the liver, these little handles reach out, grab onto the bad cholesterol boats, and pull them inside the liver to be destroyed.
That sounds incredibly efficient.
It is, usually. When it works, your liver constantly pulls the bad fat out of your blood, keeping the pipes completely clear.
So where does this system break down? Why do we get clogged pipes if we have all these vacuum handles?
The breakdown happens because of another specific protein that the liver makes.
Okay, what does this other protein do?
Well, for the sake of picturing it, think of this specific protein as a tiny biological gremlin.
A gremlin. Okay, I like that. What is the gremlin doing?
The gremlin's entire job is to hunt down your liver's vacuum handles and latch onto them.
Wait, it attacks the vacuum?
Basically, yes. Normally, a vacuum handle pulls in a piece of bad cholesterol, drops it off inside the cell, and then pops back out to grab another one.
It recycles itself.
Exactly. But if the gremlin is attached to the handle, it stops that recycling process. It essentially flags the handle for destruction, and the liver cell digests its own vacuum attachment.
Oh, wait, so our bodies naturally produce a gremlin that actively stops us from clearing out bad cholesterol.
That is exactly what is happening.
Why would evolution do that to us? That seems completely backwards.
It is a profound question, and you have to think about our evolutionary history.
Like caveman days.
Right. For most of human history, we weren't dying of clogged arteries at age 70. We were starving, or freezing, or fighting off infections.
So food was scarce?
Extremely scarce. And cholesterol is a super dense source of cellular energy.
Oh, I see where this is going.
Yeah, so having a gremlin that destroyed the vacuum handles meant the cholesterol stayed in the bloodstream longer.
Which was a massive survival advantage if you were starving during an ice age.
Exactly. It was a biological hoarding mechanism that kept early humans alive.
But now that we live in a modern era, with fast food on every corner, that same hoarding mechanism is killing us.
It is a total mismatch between our ancient biology and our modern environment.
That is fascinating. So how did scientists figure out that this gremlin protein was the culprit?
It was a massive genetic hunt that really started in France. Scientists there were looking at specific families with terribly high cholesterol.
Like off the charts high.
Yes, people having fatal heart attacks in their 20s. The scientists mapped their DNA and found a mutation.
Let me guess, their bodies were making way too many gremlins.
Exactly. Their gremlin factory was stuck in overdrive. It destroyed almost all their vacuum handles, and the bad cholesterol just built up to lethal levels.
So that proves the gremlin causes the disease. But finding a bad mutation doesn't give you a cure.
Right, it just identifies the enemy. The real breakthrough came from a huge study in Texas.
The Dallas Hines study, right?
Yes. They were just scanning the DNA of normal people in Dallas, and they found the mirror image of the French discovery.
What do you mean?
They found a small group of people who had a naturally occurring genetic topo that completely broke their gremlin factory.
Oh, wow. So they produced 0 gremlins.
Virtually zero.
And without the gremlins destroying the handles, what happened to their livers?
Their liver cells were absolutely covered in vacuum handles. They were pulling immense amounts of bad cholesterol out of the blood constantly.
So their cholesterol levels must have been incredibly low.
Staggeringly low for their entire lives.
And what about their health? Were they okay?
That was the biggest discovery of all.
Yeah.
Not only were they perfectly healthy, with completely normal brain and liver function, but they were remarkably protected from heart disease.
Like bulletproof.
Near total protection from heart attacks, with 0 negative side effects from having the mutation.
So the scientific quest became obvious. How do we artificially give everyone else that lucky, protective mutation?
Exactly. Knowing what to target is one thing, but actually going inside a living human to permanently change their genetic code is another huge challenge.
Because when most people hear gene editing, they think of molecular scissors that cut the DNA in half to break a gene.
Right, the older generation of gene editing. It works a bit like a molecular guillotine.
Wow, a guillotine. That sounds intense.
It is. It literally severs both sides of the DNA ladder.
But wait, if you want to break the gremlin factory, why not just use the scissors to chop it up?
Because relying on a double-strand cut is chaotic. When the cell panics and tries to glue the severed ends back together, it makes mistakes.
And mistakes in DNA sound like a recipe for disaster.
They can be. You can cause unwanted damage, scramble other genes, or in the worst-case scenarios, trigger cancer.
Yeah, that is a non-starter if you are trying to treat millions of otherwise healthy people for cholesterol.
Exactly. You cannot accept that level of risk. So scientists had to develop something much more elegant.
And that new method feels more like a tiny pencil and an eraser.
Right, that's the perfect way to describe it. It is called base editing.
How does base editing actually work without cutting the DNA?
It uses a disabled version of the scissor tool. It acts purely as a search engine to scan your billions of DNA letters until it finds the exact recipe for the gremlin protein.
So it hovers over the target, but the scissors are dull.
Right, no cutting. Instead, it has a chemical eraser attached to it.
Okay, so what does the eraser do?
It hovers over one specific letter in the genetic code, an A, and chemically alters it to look like a G.
Wait, it just changes a single letter out of billions?
Just one single letter swap. The outer structure of the DNA is completely untouched.
That is incredibly precise.
Right.
But is changing one letter really enough to stop the gremlin?
Absolutely. The DNA recipe is ruined. When the liver cell tries to read the instructions to build the gremlin, the typo causes the protein to misfold.
So it comes out all mangled.
Exactly. The cell's internal quality control looks at this mangled protein, assumes it is garbage, and breaks it down before it ever gets released.
The gremlin is gone forever.
The factory is permanently shut down, with 0 collateral damage to the DNA structure.
Okay, so the pencil and eraser text sounds flawless, but reading the history here, it seems like getting this tool into the liver of a living person proved to be a massive hurdle.
Delivery is always the hardest part of genetic medicine. You can't just inject raw genetic instructions into the blood.
Because the body would just destroy it.
Instantly. the immune system would shred it. So you need a delivery truck.
And in the first major human trial a few years ago, the delivery truck was a tiny bubble of fat, right?
Yes, a synthetic fatty bubble. They packed the base editor inside this bubble to protect it while it traveled to the liver.
The idea being that since liver cells are surrounded by fat, the bubble would just merge with the cell and drop off the payload.
That was the theory. And they infused millions of these bubbles into the patients.
But it caused a major setback, didn't it?
It did. While it successfully lowered cholesterol, the delivery method caused alarming side effects.
I read that one patient experienced a severe spike in liver enzymes, meaning their liver was taking damage.
And a dangerous drop in blood clotting cells, yes.
If the pencil and eraser editor is so incredibly precise, why did the patient's liver get so angry and inflamed?
Because of how the delivery truck was driving, basically.
Mhm.
It was a passive delivery method,
Meaning what, exactly?
meaning the scientists had to use a massive, highly concentrated dose of these fatty bubbles and just hope they blindly bumped into the liver cells.
So it was basically just crashing into the liver in large toxic amounts to force its way inside.
Exactly. It was a brute force approach. And your liver has specific immune cells whose only job is to detect massive influxes of foreign debris.
So the immune system recognized this massive crash of delivery trucks and went on the attack.
It triggered a massive inflammatory alarm. The liver panicked.
Which means you can't use this on the general public.
No, the safety profile was completely unacceptable. To save this miraculous cure, the scientists didn't need to change the genetic editor. They needed to build a better delivery truck.
So instead of a delivery truck ramming through the front door of the liver, they gave the truck a VIP access pass that lets it walk right through the loading dock.
That is exactly what they did for the second-generation trial that happened recently in May of 2026.
Okay, tell me about this VIP pass. How do they upgrade the truck?
They took the exact same fatty bubble, but they chemically glued a special sugar molecule to the outside of it.
A sugar molecule, how does that help?
Because on the surface of human liver cells, there's a very specific biological lock. And this lock only accepts that one specific sugar molecule as a key.
Oh, wow. So they just engineered the perfect key.
Exactly. When the delivery truck floats by, the liver cell recognizes the sugar key, grabs it, and happily swallows the bubble whole.
No crashing required.
None. Because it's so targeted, they could use a tiny fraction of the original dose. The immune system never even notices it.
No alarms, no angry liver.
Zero inflammation.
And the results from this May 2026 trial were stunning. These were tested in patients in the UK and Canada, right?
Yes, and the efficacy data is just unbelievable. From a single IV drip, just a few hours in a clinic, they saw up to an 88% reduction in the harmful gremlin protein.
Almost the entire factory shut down.
Right, and because the liver could finally grow its vacuum handles back, they saw up to a 62% drop in bad cholesterol.
From one single IV drip. And this drop has lasted for up to 18 months so far, right?
Yes, with absolutely no serious side effects. For patients who were previously facing early heart attacks despite taking max doses of pills, this is a life-altering one-time cure.
It really is a miracle. But as amazing as permanent DNA rewriting is, there's a lingering fear for some patients.
It is totally understandable. Permanently changing your DNA is a massive psychological leap.
Exactly. What if you don't want to permanently erase a letter in your genetic code? What if, I don't know, you want to be able to undo it later?
And that is why scientists are developing an alternative method right alongside the permanent editor.
If base editing is erasing a word in the recipe book, this new alternative is like slamming a heavy book on top of the page so the chef just can't read it.
That is a brilliant analogy. The scientific term is epigenetic silencing.
Epigenetic. Okay, break down what that heavy book actually is.
Instead of sending in a tool to change the actual letters of the DNA, scientists send in a tool that acts like a biological dimmer switch.
So it just turns the volume down on the gene?
It turns it off completely. It attaches chemical do not disturb signs directly onto the gremlin gene.
So the DNA isn't actually broken or changed?
No, the structural sequence is completely untouched. The heavy book just buries the recipe so tightly that the body's reading machinery ignores it entirely.
That seems like it has massive advantages for people who are scared of permanent edits.
It does. It is highly specific. It completely avoids any risk of structural DNA damage. And most importantly, it is reversible.
Reversible is the magic word there. But wait, if it's just a temporary do not disturb sign, doesn't it just fall off after a few days?
That was the worry. But the tests in monkeys proved otherwise.
What did the monkey trials show?
A single infusion kept the cholesterol suppressed for nearly a whole year.
Almost 12 months from one temporary treatment.
Yes, which means patients might only need a shot once a year instead of taking a pill every single day.
That is a phenomenal alternative. But here's my big question. Whether we use the permanent eraser or the temporary dimmer switch, these tools seem to be moving way faster than the government's ability to regulate them.
Regulatory hurdles have historically been the biggest bottleneck for heart disease treatments, absolutely.
Right. Historically, if a drug company wanted to get a heart disease drug approved, they had to run these massive decade-long trials with thousands of patients.
Yes, they literally had to wait 10 years to count how many people had fatal heart attacks on the drug versus a placebo.
But if we already know that lowering this specific gremlin protein stops heart attacks, why do we need to wait 10 years to prove it again? That seems like a waste of time and a waste of human lives.
I agree completely. And thankfully, this is exactly what their regulators recently concluded as well.
Really? They actually changed the rules.
They did. They completely changed their rule books to get these cures to patients faster, introducing something called the plausible mechanism pathway.
Plausible mechanism. What does that mean in plain English?
Regulators looked at the natural human genetics, those lucky people in Texas, and decided that the proof already exists in nature.
So if the scientists can prove the drug does the exact same thing as the lucky mutation.
Exactly. If they prove they safely turned off the target gene and the bad cholesterol drops, they don't need a 10-year study to prove it prevents heart attacks.
They can rely on the natural history of the disease.
Right. It shaves a decade off the approval timeline.
Okay, so the science works perfectly, and the government is greenlighting it at record speed. But here is the massive elephant in the room.
The economics.
How on earth do we pay for this? Because we already have cheap, generic pill statins that lower cholesterol safely.
And they cost pennies a day.
Exactly. Why would a health insurance company ever pay for a cutting-edge $1 million genetic infusion when a daily pill costs almost nothing?
If you only look at the cost of the pill, it doesn't make sense. But you have to factor in what economists call the forgetfulness penalty.
The forgetfulness penalty.
Yeah.
Because people don't actually take their pills.
Human behavior is the absolute biggest bottleneck in all of medicine.
I read that nearly 50% of people prescribed daily cholesterol pills stopped taking them within a single year.
It's staggering, but true, because high cholesterol is silent. You don't feel sick when your arteries are clogging up.
So people just skip a day, then a week, and eventually forget about it entirely.
Right. And because they stop taking the cheap pills, they eventually have a massive heart attack.
And a heart attack isn't cheap.
Heart attack costs the medical system an absolute fortune. Ambulance rides, emergency surgeries, intensive care, long-term rehab, hundreds of thousands of dollars.
Oh, I see. So the insurance companies aren't comparing the $1 million cure to the pennies for a pill.
No, they are comparing it to the massive hospital bills they know they will inevitably have to pay when the patient forgets their medication.
A one-time permanent cure completely removes human error from the equation.
Exactly. Which is why health systems are totally shifting how they pay for these therapies.
Are they using installment plans or something? You can't just drop $1 million up front.
They are looking at annuity models. It is basically like paying a mortgage over several years.
Okay, so the hospital pays a chunk every year.
Right. And they combine it with risk-sharing models.
Risk-sharing meaning the drug company is on the hook.
Yes. The drug company only gets their annual payment if the patient's cholesterol actually stays low.
Wow. So if the genetic cure wears off in year 3, the payments stop.
Immediately. It forces the drug makers to ensure these cures are actually permanent.
That is a brilliant financial model. But before we wrap up, we have to address the most profound fear people have when they hear about genetic engineering in humans.
The fear of passing it on.
Yes. Will I pass this artificial mutation on to my children? If I get this VIP delivery truck sent into my body to permanently change my DNA, is there any chance it accidentally edits my reproductive cells and changes the human gene pool forever?
It is the ultimate ethical nightmare, and it's something regulators take incredibly seriously.
So how do we know it's safe?
We have to make a crucial distinction between editing normal body cells, like your liver, and editing reproductive cells.
So body cells versus sperm and egg cells.
Right. The delivery truck is explicitly designed to only unlock the doors of the liver cells. But to prove it never goes rogue, they did exhaustive safety testing.
I want to hear the numbers on this. What kind of tests?
They gave massive doses of this exact treatment to mice before they allowed them to mate.
Okay, and then they checked the babies.
They sequenced the DNA of all 436 of their offspring.
436 babies. And how many had the genetic edit?
Absolutely zero. Not a single one of the babies inherited the edit.
Wow. 0 out of 436.
The edit is strictly confined to the body of the patient receiving it. It dies with you.
That is incredibly reassuring.
It fundamentally proves that the ethical boundary holds strong.
It really does. But I want to leave the listener with a final provocative thought to ponder.
The behavioral side of the equation.
Exactly. Because if a single genetic edit can permanently delete the risk of the world's deadliest lifestyle disease, what happens to human behavior?
It is the moral hazard of a permanent cure.
Right. If you know you are genetically bulletproof against heart disease, does it change how you eat? Does it change how you exercise and how you live your life?
Do people just start eating fast food for every meal?
Exactly. Is removing the biological consequence of our lifestyle a massive medical triumph? Or does it invite a whole new set of societal problems for us to navigate?
Science can fix the biology, but human nature is a lot harder to rewrite.
Thanks for coming along on this deep dive with us. Remember to follow us on your favorite podcast platform, and see you on the next dive.